Well, I'm always interested in where are there bottlenecks in science that could be solved by some sort of better observational tool, right? Sometimes those tools are hard to build, right? They're not something an individual student or scientist, could build. Maybe they require more engineering. And so I'm curious where that fits in here. And I'd like to ask Clarice a little bit about, I guess, what would be the sort of ideal tools for observing quantum biology in a more direct way. Like I know some of what you all have done already is just trying to validate, magnetic field effects on biological systems, but you don't necessarily know what's the mechanism of that. You just know, these magnetic fields potentially matter for something like development or, navigation in birds, I think is very much thought to be
That
like,
one's well known. Yeah.
directly magnetic field dependent. Um, but it seems like there's some kind of gap where it is hard to actually go in and do the microscopic observation of what is happening. So maybe Clarice, you could just tell me, I mean, what do you think? What is the sort of observational tool that needs to be built that would make it easier
Yeah.
to, ground truth what's going on with quantum biology? Yeah.
Okay, so in order to observe quantum biology, we need to build quantum instruments, right? So the idea is to build instruments that can perform bona fide quantum measurements inside proteins, inside cells. So, if you will, those are glorified microscopes that can look at quantum signatures inside proteins and check whether that said quantumness is being used for function, right? So I think that you know that by now that instruments drive new science. So we really need to build those glorified microscopes, and I can give you like a breakdown of what those are.
Yeah.
They're
Please.
optical microscopes coupled to coils that produce either or both DC and AC magnetic fields. Everything synchronized down to the nanosecond, because that's the time scales of relevance for the spin physics that we care about, that we think underlie magnetic field sensitivity, and with very good single photon cameras, because it's a feature of the proteins that we study, but it's also a feature of technological quantum systems that the amount of fluorescence, the amount of light that certain proteins emit is correlated with the state of the quantum state of particular electrons inside it. So fluorescence intensity is a quantum signature, is a quantum correlate, if you will. So what we would like to do in the future would be to check for how long quantumness is alive inside cells and check whether it's long enough for magnetic sensitivity to be achieved. For example, if birds truly are using the magnetic field of the Earth to navigate, the profound implication is that electron spin superpositions need to be alive inside their cells for at least 750 nanoseconds. Okay, and the smaller the field, the longer the quantumness that the cells need to support. So this is the type of instruments that we are starting to develop.
This is
and
great.
Again,
so let me see if I can just make sure I understand all this. So I'm familiar with like an MRI machine. That's more like looking at a nuclear spin. And for that, you're looking at radio waves that are kind of coming out essentially
yes.
From the MRI machine or NMR. Here, you're trying to look at the spin of electrons because you think that these are what's
Correct.
coupling between the magnetic fields and chemistry that might happen in the organism.
There's another very profound difference, because even though large magnetic fields might affect electron spins and nuclear spins in a way that people familiar with NMR and EPR are used to seeing, the magnetic fields that can tweak the fate of a broad class of chemical reactions are relatively weak. So this is a rare instance where effect magnitude as a function of field strength, it does not monotonically go up or down, it goes up and down. So that for fields which are much larger than, say, tens of times the magnetic field of your cell phone, those fields can do other things, but they cannot macroscopically influence the final products of this type of chemical reaction that is akin to quantum sensing. So this is the cool part of it, because in order to actuate on those processes, you don't need a big MRI machine, right? You can get things done with a magnetic field that is as small as that produced by your cell phone.
Okay, and then you want to look at this with an optical microscope because that allows you with the fluorescence to see, are the spins the same, or are they different, or are they staying the same over time, or how does it couple into the spin? Yeah,
different things. First of all, you can see for how long the quantum survives inside the cell. Okay, and again, you can start making checks. Oh, for inside the cells of the birds, do the spins survive in a quantum state for as long as it takes for the magnetic field of yours to be sensed? But on the other hand, you also want to try to correlate spin states to macroscopic functioning. For example, by preparing cells with different electron spins in different spin states, how does that macroscopically influence other macroscopical functionings of the cell? Because that's the profound implication of quantum biology, that at some point there is a scale shifting effect whereby tiny things happening at relatively fast time scales actually end up having macroscopic consequences on the basal physiological functioning of cells.
And despite all of the interest in, for example, how these birds are navigating with magnetic fields or some of these questions, you're saying no one has actually ever been able to directly measure the relevant spins or directly say, I'm going to prepare them in a certain configuration of spin.
Importantly, no one has proven that those things are being used for function. Okay, so weak magnetic fields have been shown across the theory of life and across different cell types to be able to up and down regulate the whole machinery of the cell, from how fast cells divide to how much energy is produced to how much DNA gets repaired, in a way consistent with this quantum model. So even though people have found magnetic fields that do stuff, I liken it to being in a haystack, grabbing a little bit of hay and finding a needle. People change biology by trial and error. What quantum biology gives you is the predictive power on how to engineer those fields to do things. And in order to get there, the first thing is to make sure we can prove unambiguously that those things are being used for function inside cells.
And so I guess I have a couple questions about this. So one is, what's so hard about doing this? I mean, you have microscopes that measure fluorescence. You don't need that big of magnetic fields to put on the thing. What is it that makes this like especially technically challenging to measure these spins inside a living cell?
You need to control for many different things, right? For example, if you want to study the effect of weak magnetic fields, you probably need to have some sort of hypomagnetic chamber to protect your experiment from someone arriving with a cell phone in their pocket or so forth, right? You need that. You need a way to keep the cells happy because conventional microscopes like the ones one can build to look at technological quantum sensors, they're not gentle on cells, so you need to improve that part too. So it's really bringing together the experimental quantum physicist toolkit with the biologist toolkit, and those two things need to go hand in hand.
And then do you have a clear sense of what are the sort of the first spins that you want to look at? Are they in a certain protein? Are they in a protein that you are engineering? Like I've seen this recent magnetogenetics work with GFP or engineered
correct
proteins.
I
just
think
look at a tadpole. Or, how do you find the spin? Yeah.
There are broad classes of protein that are known to be magnetosensitive. People cannot predict which ones are going to be magnetosensitive, but people usually find out that redox biology related proteins are magnetosensitive, like the proteins that are involved in the give and take in electrons, and there's a reason for that. So whereas I find it very nice to have engineered proteins put on the cell to be able to perform functions upon a magnetic field, I think that's a little bit missing the forest for the trees, because the important thing here would be to be able to use the native proteins as a lever to accomplish something in biology. Quantum biology would give you like this whole different lever that is already there, You don't need to genetic engineer anything. You would just need to learn how to control each particular protein. And the idea is that each particular protein responds to a very particular combination of magnetic field intensity, frequency, and direction, right? So if you could find this code book that we're calling, if you could find this code book on how to alter specific proteins function using those exogenous, easy to produce magnetic fields, you gain.
And do you think they're going to be different for the different proteins? Like what if we're
Of
seeing,
course,
effect
of
on
course.
tadpole development, is that just kind of all this, the magnetic field is just doing 10 different things to 10 different proteins, or could you dial into a specific one, or do you think that the biology dials into a specific one?
So far, I think that people have seen broadly unspecific effects, exactly because effects are coming from many different things. But I do think we can dial in precisely. For example, I expect the same protein in the nucleus to react to slightly different magnetic fields if the protein is in the cytoplasm, because the local magnetic field environments of those proteins are going to be different, and that's going to influence the external magnetic fields that we will need to apply to be able to shift the chemical reaction. So yes, right now that's not done, but that's sort of the predictive power that we aim to get from our particular quantum instruments that we want to develop. That's the goal of it. That's the power of it.
And I always like to also think about, please go ahead.
Yeah. And I think that for someone with an interest in BCI like yourself, we have a profound hypothesis too, which is the fact that maybe ion channels inside neurons or inside all cells, maybe those might be producing transient magnetic fields that are finely tuned to affect proteins that respond to magnetic fields in different parts of the cells, right? Ion channel fluxes vary by duration, by strength of the flux. So I wonder if those are not being utilized by the cell as the tuning fork for this type of electron spin dependent chemical reaction.
Yeah, that's really interesting. I mean, I always also like to think about, there's the most ambitious version and there's sort of fallback versions. So even if you did have to genetically engineer the cells, I mean, there's still things that you could do. You could engineer your own quantum sensor, right?
Yes.
Even if they're not easy to dial into a particular thing.
Yes.
Endogenously, right?
Yes, and I think that is a useful first step. And I think we cannot lose sight from the fact that the degree of freedom is there.
Cool. And yeah, I mean, what does the effort look like? So is this physicists, is this engineers, biologists? Like what does it look like to do? I don't know where it is on your roadmap, but
we don't have stuff on the team on there, but we have our current team, and then we have who we're planning to hire, like essentially lined up. But we have like Clarice is obviously the lead. We have Alessandro, who is the he does experimental quantum physics and does the magnetic side. And then we have a microscope, optical microscope. Is that the right way to describe it? Anyway, optical microscope designer, physicist, and then you should actually describe the titles here, Clarice. You have a better grip. What are the two types of biologists that we're getting?
So it's one who can do protein engineering and the other one who can do cell and molecular biology. So the team needs to be interdisciplinary by design, right? Otherwise, you cannot advance quantum biology. And I think that's both the coolest thing and the most daunting thing about quantum bio. But we need to have a very close collaboration between people with different backgrounds.
And you think you can do some of this in like more isolated cells versus in, maybe
I think
bird
that's
has a
the
whole organ or something that's using for, finding the magnetic field.
So I think that in the beginning, we will need to do this in a cell because things are more controlled, right? We need to build from the ground up because right now there's a big chasm, a big gap between experiments in proteins in solution and experiments in plates of cells or organisms. We really need to build all the missing steps between the two because it's going to be a daunting task to go from magnetic field sensitivity to function to even more to behavior, as in the case of birds. And again, birds are just the poster children of this field. There's a lot of evidence that this is way more prevalent than just birds, that it's permeating the tree of life, if you want to see what we think. We think that there is this bubbling quantum bath that is actually underlying how every cell in your body behaves.
And you said something, to detect certain strength of magnetic field, it needs a certain amount of nanoseconds of the spins sort of being coherent, or
that's correct.
How do you measure this tiny number of nanoseconds, and how sure are you about how, about this question of how long it has to stay coherent?
I spent my whole thesis, and people spend their whole thesis like writing about ways to estimate coherence times, the quantumness times. But back of the envelope calculation is as follows. In the presence of a magnetic field, a spin will Larmor precess. We can say that a spin is sensing a magnetic field if it can Larmor precess once before losing their quantum character, because it's this Larmor precession. Actually, it's a spin superposition that is going to be able to sense the magnetic field once the different components of the superposition processes. So in order to sense the magnetic field of the Earth, an electron spin superposition needs to be alive for the time that it takes for an electron spin to process around the magnetic field of the Earth, which is 750 nanoseconds. But in order for a spin to sense the magnetic field produced by your cell phone, which is about 1 millitesla, the coherence time is only 50 nanoseconds. So even if super long coherence times are not available in nature, maybe we could already use short coherence times to sense.
And so just so I'm understanding, so the cell phone field is actually a lot bigger than the Earth field.
That's correct.
And that's why. Okay, that's great. Yeah, I guess that's great. Anyway, I don't tend to worry about this, but maybe I should. Anyway, no, this is really
here's what we tell people. Because every protein reacts to a specific band of magnetic field frequency, intensity, and duration, I tell people that it's very unlikely that your cell phone is actually doing something, or at least it's as unlikely
it's all
that
averaging
it's doing
out,
something.
Kind of, yeah.
No, it's just that it's unlikely that it's doing something bad for you, as unlikely as it is that it's doing something good for you, right? But it doesn't mean that, as you said, we cannot learn how to engineer those fields for function.
Super cool. No, this is super, super interesting. And how long do you think it will take you to build this microscope, or to do these experiments? Yeah.
We have it's on our roadmap, but we have 2 years for the construction of the microscope.
Okay.
And I am still optimistic. We've tried some. We have not shortened the time frame yet, but I'm optimistic we'd be able to do it faster. And then the plan is concurrent with the construction of the microscope to develop the slate of proteins that we want to check, because we're going to check basically the more easily controlled things first. So you do proteins outside of cells, then you do proteins inside cells in a protected area, then proteins inside cells, and then
are you going to use these magnetogenetics proteins that are coming out? There have been some measurements on GFP, and there's also been, I guess a mutant that is magnetofluorescent. Yeah.
So yes, and I think those are great. I think those are very nice steps towards
positive
doing
control
something
for what you're doing, or something, maybe, or
yes. And I think that even though we're going to take that as a first step, I repeat, the goal here is to use what is already in your cell.
Right. Yeah, that makes sense.
Yeah, the engineer.
Well, if I was a jellyfish, I guess, or maybe I would already have my GFP. Anyway, that's great. Yeah. So okay. Yeah. Thanks.
But all your cells have, for example, flavoproteins. Flavoproteins respond to magnetic fields. You don't need to genetically encode anything, right? Or the tadpole cell, right? We don't know how that's responding. The most likely explanation is that it's quantum, but this shows that the lever is there, that the button is
And
there.
are you all searching for mutants of these tadpoles that would like maybe not respond to the magnetic field in this way, or like to try to narrow down on what the proteins are causing it? Or
we have started looking. We met a biologist who looks like they might be quite good at helping with this sort of experiment, and we were just at the beginning of trying to think about what's possible here.
And the tadpole experiment was conceived to update the scientific consensus that those effects are both real and measurable. Right? Even there are websites from US government that says, no, weak magnetic fields don't do anything to you because they're too small, they don't trigger a biological mechanism. And that's misleading, that's false, right? The tadpoles were chosen because of how common they are. There's nothing particular about tadpoles. We bet we would find similar effects in any other model organism that we picked. In the experiment, all the analysis, all the images are out, and it's just there to update the consensus that these effects are real. We controlled for light, temperature, humidity, evaporation in a way that hasn't been controlled before. So it's very hard for people to say that it's an experimental fluke. We looked at more than 8,000 images, and it's also very hard for people to say that we are not controlling.
And it's up to date. They can talk to you. It's not just something that's buried in the literature somewhere. Yeah. No, this is great. Okay, awesome. Well, hopefully this is a good science conversation for you guys. I don't know. And so,
works for us.
Sort of biophysics conversation. So awesome.
Perfect.
Yeah, great.
Thank you so much. All right.
You're welcome.
See you. Yep.